Welcome back to the deep dive. This is where we take complex research and really break it down for you. Today, we're diving into biological invasions in the ocean. Those species ending up where they didn't start out. And, the really key thing here is that it's us, human activity, ships, aquaculture, all that stuff that's driving most of this movement around the globe.
It's a huge issue. Ecologically, economically,
Hmm.
it's causing real problems. But the way we've traditionally tracked how these species move, it's been, well, pretty fragmented, kind of imprecise, scientifically speaking.
Yeah, and that makes managing it almost impossible, doesn't it, if you can't clearly define the mechanism?
Exactly. You can't manage what you can't measure or even clearly describe.
Which brings us to the focus of today's deep dive. It's a really important paper, Anthropogenic Vectors of Marine and Estuarine Invasions, an overview framework. The authors are James T. Carlton and Gregory M. Ruiz.
A foundational piece, really.
Our mission today is basically to unpack the framework they propose. It's quite rigorous, has 6 parts. And the idea is by the end of this, you'll get the toolkit they've built for analyzing how species get moved around passively. It's all about that first step, the transport stage in the whole invasion process.
Yeah, because before this paper, the terms used were just, well, often too fuzzy. Made it hard to create effective management strategies.
So what did Carlton and Ruiz do? What's the core idea?
Their big move, the really key thing, is they argue we should ditch the term pathway. It's just too vague, right? People use it for everything from the ship itself to the entire ocean route.
Okay, so ditch pathway, what do they replace it with?
They break it down into 6 specific, distinct concepts. The aim is to bring much-needed clarity, especially for regulation and management.
6 concepts, okay. And this isn't just like semantics, it actually changes how we look at invasions.
Oh, absolutely. It forces a much more structured way of thinking. It covers all the stages: transport, introduction, establishment, and spread. But let's start with the first 4, which really define the physical movement part, replacing that old pathway idea.
All right, let's get into the nuts and bolts. What's the first element?
Okay, so the first one is cause E. Simple question. Why is a species being transported?
Why? Okay. Like on purpose or by accident?
Exactly. They categorize it as either accidental AC, so unintentional hitchhikers, maybe in ballast water or stuck on a hull,
Uh-huh.
or deliberate DB. That's when it's intentional, maybe for aquaculture or stocking a fishery, something like that.
Okay, accidental or deliberate. But I can imagine looking back, especially for older invasions, telling the difference must be incredibly hard sometimes. How do you actually quantify that?
Yeah, it's definitely tricky. The quantification involves sorting species into AC, DB, or, importantly, both.
Both. So a species could be moved deliberately and accidentally?
For sure. Happens all the time. A species might be intentionally introduced for farming. Maybe it fails to establish then, but later it gets picked up accidentally by ships and spread that way.
Ah, go on.
So unless you have really good historical records, or you can do like detailed genetic analysis to trace the origins, figuring out the original cause can be tough.
Huh.
You often need multiple lines of evidence.
Got it. So cause is the why. What's next? Tracking the journey.
Precisely. Element 2 is RUT. This is the geographic path the transport takes. So from the donor region, often defined by the last port of call, the LPOC,
POC, last port of call. Okay.
all the way to the recipient region where it arrives.
And how do you quantify a route? Is it just like drawing a line on a map?
It's a bit more than that. You need to know the number and location of all potential donor and recipient regions along that path, and also the range of different geographic or environmental regimes it passes through. Like, does it stay in tropical waters or go from temperate to tropical, that kind of thing?
Right. Okay, let me pause on that last port of call idea. Say a ship goes from, I don't know, Rotterdam to New York, but it stops for fuel quickly in Halifax. Is Rotterdam still the LPOC, or does Halifax muddy the waters?
That's a great question, and it shows why the framework needs to be rigorous. Usually, the LPOC is the last place the vessel had significant interaction, like taking on ballast or spending enough time for things to attach or fall off.
So a quick fuel stop might not count.
Might not, unless there was significant water exchange or something. But the key is the quantification should track all potential donor regions. So Halifax would still be noted as part of the overall route, even if Rotterdam is the primary LPOC. It captures that complexity.
Okay, so route is the big journey. Then what's corridor? You mentioned that one too. Is that like a specific channel within the route?
Exactly. Think of it as the physical conduit within a specific route. The classic examples are things like canals, the Suez Canal, the Panama Canal. Major shipping lanes could also be considered corridors.
So man-made structures or heavily used paths, like a highway on the ocean.
That's a good way to put it. On land, you might think of a highway itself, or a railway line, as a corridor facilitating movement along a route.
Okay, so route is the general path, corridor is the specific conduit. How does that differ from the actual thing doing the carrying?
Ah, that brings us neatly to element 4, the victor. This is the how, the actual physical means or agent that transports the species.
Right, so the ship itself.
The ship, definitely. But also its subcomponents, the hull, the anchor chains, the ballast tanks. But it's broader than just ships. It includes things like dry docks being moved, contaminated aquaculture stock, live seafood trade, even fishing bait.
Wow, okay. Lots of possibilities.
Oh yeah, marine debris is a big one now, plastics, styrofoam floating across oceans.
Huh.
And they even include niche things like species hitching rides on tracking bands put on birds or marine mammals or, get this, scientific sampling gear being moved between sites.
Scientific gear, that's kind of ironic, isn't it? We study the ocean and accidentally move species around with our tools.
It really highlights how pervasive these human-mediated movements are. And importantly, when you quantify the vector, it's not just what it is, but its characteristics.
Like what?
Things like its size, the surface area of a hull, for example, the rate how much ballast water is discharged over time, the duration of the transit, how long the journey takes, and timing like the season, which affects survival.
Okay, so transit time matters. Faster isn't always better for the hitchhikers, maybe.
Often, yes, shorter transit times can mean less time to reproduce en route or higher mortality for some organisms. So it's a critical variable for assessing risk.
Right, so we got cause, why, route, the path, corridor, the channel, and vector, the vehicle means. So that maps out the physical journey. But does knowing a ship arrived tell you if anything dangerous actually got off?
Exactly. That's the crucial next step. Just knowing the physical transport happened isn't enough. You need to measure the biological payload, which leads us to the fifth element, propagule pressure.
Propagal pressure sounds important. What is it exactly?
Think of it as the inoculum. It's about the organisms themselves. It covers three things: the quantity, how many individuals arrive; the quality, are they viable, healthy, able to reproduce; and the frequency, how often are they arriving.
Okay, quantity, quality, frequency. Makes sense. A single arrival of 1 million larvae is different from one larva arriving every day for years.
Precisely. And the authors point out a massive challenge here. Those traditional metrics we mentioned, like just counting ships, they can be really poor indicators of the actual propagule pressure.
Why is it so hard to measure the inoculum accurately?
Two main reasons, really. First is what they call taxonomic limitation.
Meaning we don't know what all the species are.
Pretty much. We tend to focus on the bigger, more obvious stuff: crabs, mussels, fish. But the tiny things, microscopic protists, nematodes, tiny worms, phytoplankton, they often get ignored or just lumped into broad groups because identifying them is hard, requires specialist expertise.
So we could be missing a huge chunk of the picture, like ignoring all the small cars and bikes and only counting the trucks on the highway.
That's a perfect analogy. We're potentially vastly underestimating the sheer diversity and number of propagules arriving just because we're not looking for or can't easily identify the small stuff. And those micro-invaders can have huge ecological impacts down the line.
Wow. Okay. So taxonomic limits. What's the second challenge?
The second one is viability quality. Even if millions of propagules arrive, it doesn't matter if they're already dead or too stressed to survive and reproduce.
Right. They could get cooked in warm ballast water or shocked by different salinity when released.
Exactly. They need to be physiologically capable of handling the new environment after they arrive.
So how do researchers figure that out? You can't just count them. You need to know if they're alive and kicking.
This is where experimental work becomes crucial. Scientists will actually take samples, say from ballast water, and run tests. They'll check the survival rates of specific organisms like crab larvae under conditions mimicking the destination port, the temperature, the salinity.
Like seeing if larvae from the tropics can survive in, say, Chesapeake Bay water.
Precisely. Or they'll test phytoplankton using special stains or trying to culture them in growth media to see if they are viable and can actually start a new population.
And what's the takeaway from those kinds of studies? Do things survive?
The general conclusion, based on quite a bit of work like that cited in the paper, is yes. Despite the journey being tough, a surprising number and variety of organisms do survive transport in vectors like ballast water and are capable of establishing. The pressure is definitely real.
Okay, so the pressure is real and often underestimated. This leads to the final element, right? The outcome.
Yes, the sixth element is vector strength. This is basically the bottom line. It measures the success rate of a vector over time.
How is it defined?
It's the number of established non-native species that can be attributed to a specific vector in a specific region over a certain period. So if ballast water is linked to, say, 50 successful invasions in San Francisco Bay over 30 years, that gives you a measure of its strength.
Okay. But you mentioned earlier it's hard to pin down because of poly something.
Polyvectism, yeah. This is a major complicating factor and why vector strength is usually underestimated.
And polyvectism means?
It means a single species is often transported by multiple vectors, not just one.
Ah, right. So that clam you mentioned could arrive on ship hulls and in ballast water and maybe with imported oysters.
Exactly. All at the same time, or maybe consecutively.
Right.
When that happens, trying to figure out how much of the blame or strength belongs to just the ship hulls versus just the ballast water
Mm-hmm.
becomes incredibly difficult, almost impossible sometimes. It really muddies the picture for management.
Okay, this framework, cause, route, corridor, vector, propagable pressure, vector strength, it makes a lot of conceptual sense. But how does it play out in the real world? The paper gives some examples, right?
Oh yeah, and some of the anecdotes really bring it to life.
Yeah.
They show how thinking about routes and corridors specifically helps understand weird dispersal events.
Like what?
Well, there's this fascinating aspect of route accumulation. The idea that a vector, like a ship, can actually pick up new species along its journey.
So it doesn't just carry things from point A to point B, it can collect passengers at point C and D too.
Exactly. Charles Darwin even observed this way back in 1854. He described a ship going from Britain towards West Africa. It picked up one species of barnacle, then it sailed to South America, and a different barnacle species settled on top of the first one.
Wow. Layered hitchhikers.
Yeah. And then, on the way back, it picked up more of the first barnacle species, settling on the second layer. A multi-stage accumulation over a massive geographic route.
That's amazing. And this still happens.
Absolutely. Modern studies confirm it. And think about marine debris, all the plastic and junk floating around now. It lasts for ages. That debris acts like a mobile route, constantly picking up and dispersing species.
Like rafts carrying coastal species across entire oceans.
Tragically, yes. The prime example is the debris from the 2011 Tushoku tsunami in Japan. Huge amounts of wreckage docks, buoys, household items floated across the Pacific.
Right, I remember hearing about that.
And they carried hundreds of Japanese coastal species, alive, all the way to North America and Hawaii. That debris field acted as a massive unintended vector, demonstrating route accumulation and showing polyvectism in action. Species normally moved by ships suddenly had this whole other way to travel.
That's sobering. And what about corridors? Any examples where the specific channel mattered in an unexpected way?
There's this fantastic story about the Suez Canal, a major corridor involving the Yellow Fleet.
The Yellow Fleet? What was that?
During the Six-Day War in 1967, the Suez Canal was blocked. 14 large cargo ships got trapped inside the Great Bitter Lake, which is part of the canal system,
Trapped for how long?
for 8 years, until 1975. They were stuck there, covered in desert dust, hence the nickname Yellow Fleet.
8 years stationary in a canal lake. What happened biologically?
Well, sitting there for that long, they essentially became artificial reefs. They accumulated what was described as a massive biofouling aggregation, a whole community of organisms native to that part of the canal system.
Okay, so then what happened when they finally left?
Before they sailed out, scientists actually sampled this huge buildup of life on their hulls. When those 14 ships eventually moved out into the Mediterranean or the Red Sea, they potentially dispersed that entire dense community of species they'd acquired during their long stay in that specific corridor.
Wow. So the corridor itself, because of that unexpected event, became an incubator and then a launch pad for potential invasions.
Exactly. It shows how understanding the specifics of corridors, not just the overall route, is vital.
This really drives home how this framework isn't just academic. It has real implications for how we manage these risks, right?
Huge implications. If regulators can clearly distinguish between the cause, the route, the corridor, and the vector, they can target interventions much more effectively.
Like focusing on high-risk corridors or specific parts of a vector, maybe? Like tackling hull fouling separately from ballast water?
Precisely. It allows for much smarter, more targeted management and enforcement rather than just, broad strokes.
And we have to layer something else on top of this whole framework now, don't we? Climate change.
Absolutely critical. The authors and later work emphasize that virtually every single one of these 6 elements is going to be affected by global climate change.
How so?
Well, warming sea temperatures might change survival rates during transit. maybe making it easier for some species to survive longer journeys. Sea level rise changes harbor structures and depths, affecting vessel traffic and corridors. Changes in storm frequency and intensity could dramatically increase the role of marine debris as a vector. It affects everything.
So any future risk assessment using this framework has to account for climate change impacts.
Definitely. It's a fundamental overlay now.
Okay, so the framework is powerful, it's relevant, but the paper also points out a pretty major limitation, a big gap in our knowledge.
Yeah, this is quite striking.
Yeah.
Despite all the research and invasion signs, they state that as far as they knew when writing, there wasn't a single port, harbor, bay, or estuary anywhere in the world where all 6 elements have been comprehensively studied together.
Not one. Nobody has looked at the cause, route, corridor, vector, propagule pressure, and vector strength, all in one place, historically and currently.
Apparently not, at least not in a fully integrated way. We have pieces of the puzzle from many places, but not the complete picture, according to them.
That's a huge knowledge gap.
It really is. But it also sets a clear target for future research, doesn't it? The goal has to be to actually characterize all 6 elements, ideally in multiple locations, to truly understand the total vector pressure and, frankly, to fully test the utility of this framework in a real-world setting.
So to wrap up, the real power of this Carlton and Ruiz framework is moving us away from that fuzzy catch-all term pathway and giving us this precise 6-part toolkit: the why, cause, the geography, route, corridor, the how, vector, the amount, propagule pressure, and the result, vector strength.
It forces a level of detail and rigor that just wasn't standard before. It provides a common language and a structure for tackling a really complex global problem.
Which leaves us with a final thought, perhaps something for you, the listener, to consider.
Yeah, if we accept what the evidence strongly suggests, that polyvectism, species moving via multiple vectors simultaneously, is actually the norm, not the exception, what does that really mean for management?
Meaning, if you regulate just ballast water.
Right. You close off one vector, but the species is also arriving via hull fouling and maybe aquaculture imports.
Right.
And have you actually solved the problem? It implies that tackling invasions effectively might require managing multiple vectors concurrently, maybe even like coordinated vector blitzes.
So the question becomes, how comprehensive does our approach need to be? Given the sheer complexity and the number of ways species can move, how many fronts do we need to fight on simultaneously to genuinely slow down this global reshuffling of marine life?
It suggests the need for a truly holistic, integrated strategy. Thinking about just one vector in isolation might not be enough anymore.